I did grade your exams and I posted the grades on Blackboard, so if you want to know your grades, you can check it there. I will pass back the exam, but I'm gonna wait till next week to do so. So if you want to like look at your exam, I promise I'll give it back to you. It'll just happen next week, okay? Second of all, we talked about the absence policy or the attendance policy on the first day of class, but I'm gonna go over it again because everybody and their mom is emailing me.
Mm-hmm.
So if you miss a class, you are counted absent regardless of if you make it up otherwise. So if you're in a Monday lab and you miss it, but you end up going to Wednesday lab or vice versa, you can't. If you ask me, can I go to another lab to make it up? Yes, the answer is you can always go to another lab, but you will still be counted absent because you are not present in your registered lab time, okay? So for those of you who have been attending other classes, that is fine. I'm completely okay with you going to other labs and doing whatever, but I'm still gonna count you absent because that's what the TCC policy is, okay? Now, having said that, if you have any absence at all, one absence is 2, 3, 4, 5, whatever. Anytime I log an absence in WebAdvisor, TCC, the like automated system, sends you an email that says that you're warning you that you're like almost 15% or whatever in violation of whatever that warning is. Yeah, and you're gonna be dropped. No, you're not gonna be dropped. That's not sent by me. That's sent by the school. It's automated. If you are in danger of being dropped, I will personally talk to you myself before I drop you. And I'm too lazy to do that, so you're not gonna drop me from this class, okay? So no worries. It doesn't matter how many absences you have, you're fine, okay? So I just want to get that out of the way because everybody has been emailing me about absences. I promise you're okay even though you have it like stacking up against you if you're attending other labs, okay? Any questions on that? Okay, I took some medication for my flu thing and I'm feeling really lightheaded and I can barely see you right now. I'm like almost seeing double and I can't breathe. But we're gonna go through this, so if I don't make sense, then just raise your hand and be like, do that again. And I'll gonna try to work through it. All right, so today we are starting the blood vessel portion of the cardiovascular system. To be honest, if you look at this chapter, halfway through the PowerPoint, so about slide 42 right here, this is where we start getting into the actual blood vessels themselves. And if you were in my lab on Monday, we drew them out on the board, which is the exact same thing that these diagrams are. If you're in lab tonight, we'll do the same thing. So I probably won't draw them again just because you're already doing it once in lab. So if in reality, this chapter is only 41 slides long, instead of the 69, that's however long this is. So we should be ahead of schedule. So I've already posted chapter 20. So just in case we're running ahead, I will, this weekend, I'll post chapters 21 and 22. But I'm just letting you know that we might run ahead if you want to go ahead and print out the slides for the next few chapters after that, okay? Just in case. Who knows, I might get long-winded and get behind.
Our exam day's not gonna switch. That's gonna stay that Wednesday, March 4th.
Say that again?
The exam 2 is still gonna be on Wednesday, March 4th,
Yeah,
no
so
matter how ahead you get.
Yeah. If we get way ahead, then we'll just have a review on the last day. So it just kind of depends on the pace. Oops. Okay. So let's start into the blood vessels. There are 3 major categories of vessels. You have something called arteries, capillaries, and veins. Arteries, as we have mentioned before, carry blood away from the heart, and I like that arteries begins with with an A, and away begins with an A, arteries away. Veins carry blood to the heart, okay? So it does the opposite. Capillaries are in between, and this is where gas exchange happens. So I want to point out that if this is the heart right here, and you have a blood vessel coming out of the heart, so this is your artery. You have a blood vessel going into the heart. This is your vein. The capillary is in between these two vessels right here. This is your capillary. This is where the actual, what we call gas exchange happens, okay? So if this is your tissues right here, oxygen goes into the tissues through the capillary, carbon dioxide leaves the tissues through the capillary. So the fact that this is entering and exiting the blood vessel, this is gas exchange, okay? Gas exchange is not the same thing as gas transport. Transport just means going from point A to point B. So the fact that this is going from the heart to the tissues, this artery is responsible for gas transport. This vein is where oxygen is going from the tissues back to the heart. That's also gas transport. So transport is just going from one point to another. Exchange is where it's actually going through the blood vessel into the tissues itself. Does that make sense? So the reason I'm saying that is a lot of people think that gas exchange happens in your arteries and veins. It does not. It only happens in the capillaries here, okay? Now, we've discussed before that arteries are oxygenated in your systemic system, deoxygenated in the pulmonary, and then it's opposite in the, your veins are deoxygenated in the systemic and then oxygenated in the pulmonary. So that's just going back to that circulation thing that we did in the heart chapter, okay? So you can see how important it is from the heart chapter that we're doing it again. We're gonna also see it in the lymphatic system, okay? So this is just a diagram showing you that you have your heart, arteries with the blood coming out, capillaries is where gas exchange happens, and then veins is where the blood returns to the heart. So those are general categories of vessels, but in reality, there's more specific types. So we have different kinds of arteries, different kinds of veins, and different kinds of capillaries. So that's what we're gonna look into now, okay? So, first things first, we mentioned that all hollow organs in the entire body, whether it's your heart, your blood vessels, your stomach, your intestines, your bladder, whatever, they're made of layers in their wall, okay? What were the 3 layers of the heart? What was the innermost layer of the heart called?
Endocardium.
Endocardium. And the middle layer? Myocardium. And the outer layer? Epicardium. Okay, so 3 layers in the heart. We're going to continue with 3 layers in the blood vessels as well. I want to point out this is blood vessels. This is not the same thing as your heart. Okay, so the innermost layer is called the tunica intima. The word tunica means tunic, which is like Latin, German, one of those, for layer. Okay, so it's saying inner layer. This is going to be made out of simple squamous epithelium. So if you remember, simple squamous epithelium just means a single layer of flat cells. Now, similar to the heart, its purpose is not diffusion and filtration like it would be normally elsewhere in the body. Its sole purpose is to provide like a smooth lining on the inside of the vessels to prevent blood clotting and just reduce friction. Okay. Now, the middle layer, if you remember the middle layer in the heart was the myocardium. It was made out of what type of tissue?
Cardiac muscle.
Cardiac muscle, okay? Here, the tunica media is also made out of muscle, but we switch from cardiac to smooth muscle, okay? Now, any organ that is hollow is gonna have smooth muscle. It occurs in 2 layers. So if this is your vessel right here, you have what's called the inner circular layer, where the smooth muscle cells are arranged in that they go around the circumference of the vessel, so they go in this direction. The outer layer is what we call the longitudinal layer, and it goes along the length of the vessel. So while these are going in a circle, the outer layer goes along the length this way, okay? So what that does is whenever this layer contracts, it controls the like the diameter, how big or small it is. This right here, when it contracts, it's like an accordion. It controls the vessel length. Okay, so they work together to control blood flow within that blood vessel. Now, specifically, when we narrow this, the center right here, that's called vasoconstriction. When we widen it, it's called vasodilation. Okay, so the fact that it changes its vessel diameter is due to this smooth muscle layer. Okay. Now, similar to the epicardium, that outermost layer is going to be made out of connective tissue, like a loose connective tissue, and its purpose is basically to anchor those blood vessels to the surrounding structure so that it's not like rolling or moving around everywhere. It just kind of holds it in place. Okay, so those are the three layers right here. So the different vessels that we're going to look at, the different types of arteries, the different types of veins, and all that, we're going to look at how these layers right here differ between between the different vessels. Okay. So let's start with arteries. We have three different types of arteries. The first is called an elastic artery. This is going to be your largest of all of them. They're the largest because they're the ones that are closest to the heart. Okay? So what artery is coming out of your left ventricle?
Aorta.
Your aorta, which is the largest vessel we have in our body. What vessel is coming out of the right ventricle? Pulmonary trunk. Both of these vessels and the vessels immediately coming after that, they're all going to be elastic arteries. Now that tunica media, instead of having a lot of smooth muscle, is going to have actually more elastic connective tissue, like the name implies. So think back to AMP1 with elastic connective tissue. What's that function? Stretch and recoil, right? So basically, if your heart is right here, and this right here we said is your left ventricle, look at the size of the left ventricle, and we said that the vessel coming out of it was the aorta. Look how small the aorta is in comparison to the size of the ventricle. If this is full of blood, then as this blood flows into this vessel, it has to stretch to accommodate that volume. Does that make sense? Okay? Now, as this blood is flowing through, as soon as it passes out of this vessel into the next vessel, we want this to go back to its normal shape. So it's gonna stretch out and recoil, stretch and recoil every single time that heart pumps. Okay? So the vessels closest to the heart are going to be your elastic. The further away we move from the heart, we're going to transition in that tunica media. We're going to start replacing that elastic fibers with more of your muscular, like your smooth muscle. So those are now called your muscular arteries, and these are the ones that are going to vasoconstrict and vasodilate. So the majority of the vessels in our body that we think about and name and talk about, they're going to be your muscular arteries. So for example, we have your carotid artery, and we have your brachial artery, and your radial artery, your femoral artery, your tibial artery. All of the arteries in our body that are named are gonna be mostly these arteries, muscular arteries right here, okay? Now, we said that arteries lead to capillaries. Your capillaries are very, very, very small, okay? They're like microscopic, they're only large enough to let one red blood cell through, okay? So if you imagine how small the red blood cell, if you imagine how small the capillaries are, the arteries leading to that have to get smaller and smaller and smaller. So the smallest artery right here, before we even get to the capillary, this is gonna be your arteriole. Okay, so it's just the smallest type of artery that leads to the capillary bed. Now, if you're an arteriole and you're on this end, you are still gonna have the three layers. You're gonna have tunica media, tunica intima, media, and externa, right? But if you're an arteriole and you're closer to the capillary end, you're gonna have lost most of the layers. The only thing that it will be composed of is that tunica intima, which is a simple squamous epithelium, and you may have like one or two smooth muscle cells wrapped around it. Not a whole layer, just like one muscle cell or a muscle cell here, which controls like the vasoconstriction and dilation in that small tiny vessel, okay? So that's what this is saying here. Larger arterioles are gonna have all three layers, smaller ones only have that intima and maybe some smooth muscle. So I think this right here is showing you, I'll come back to the other slides in a minute. It's just showing you the different layers. You can see that you have the tunica intima here, which is the simple squamous epithelium. Then you have your, what's it called, smooth muscle right here, which is your tunica media, and then it's connective tissue, which is your tunica externa. As we get closer to the capillary bed, it gets smaller, and you can see that it's only that inner layer at this point, okay? Any questions on the arteries? Okay, so the next type of vessels are the capillaries right here. And we said that this is where gas exchange happens. Now, gas exchange is what their capillaries are mostly known for. But keep in mind that, like when we were talking about blood, like gas is not the only thing in blood, right? We had proteins, we had hormones, we had electrolytes, we had white blood cells, red blood cells, all the different things in blood. So all of those other components, depending on what type of capillary bed we have, are going to be able to or not be able to go into the tissues, okay? So we have three different types of capillaries: continuous, fenestrated, and sinusoid, which are on this next slide right here. The first type is the continuous. This is the most common type of vessel, or most common type of capillary bed. If you look at the word continuous, it's saying that literally every single simple squamous cell is touching each other, okay? So you have a simple squamous cell right here. It's literally touching that next simple squamous cell there, next simple squamous cell there. So if you have a molecule that wants to pass through this barrier right here, it has to be very, very, very small, okay? So only the smallest particles can pass through here, which is your oxygen and your carbon dioxide molecules, okay? Now, the second type of capillary is called fenestrated. And you can see here that it has these what are called fenestrations. There's these holes in the vessel or in the cell itself. But in addition to that, you have more like bigger gaps in between the cells themselves. So right here we said that you had a simple squamous cell and they're all touching each other. When we have fenestrated, the cells are going to be a little spaced further apart from each other, But also, notice that this is like uniformly thick right here, okay? When you have a fenestrated capillary, what happens is you have a cell that would kind of thin out in certain areas, or if it thins out completely, sometimes it makes holes, like the picture you can see right there. These are gonna allow like bigger molecules to pass through. So you're gonna have another cell right here. Ooh, I can't draw today. So can you guys see how there's like these little indentations in the cell? Okay, so between the indentations and the little gaps right here, larger molecules like glucose, for example, and other nutrients, these can now pass through easier. Glucose is too big to pass through the continuous, but it can pass through these fenestrated. Does that make sense? So it's just like making the gaps a little bit bigger. So that means that this last type of vessel, the sinusoidal capillary, these are going to have the largest gaps. So you're going to see that you have a cell here, you're going to have a cell here, you're going to have a cell here, another one over here. You can see that there are huge gaps in between these vessels, and that's going to allow large molecules like proteins to pass through where they normally wouldn't be able to. Okay? So, and like also like blood cells, so like if this is a red blood cell, you normally would want to contain it inside your like blood vessel, but for example like in the spleen, we talked about the red blood cell dying in the spleen, right? Okay, so in the spleen, what happens is this red blood cell, since it's on its way to die, it exits the blood vessel, goes into the splenic tissue where the like macrophages like go to it and basically eat it and get rid of it. Okay? Any questions on the 3 different types of capillary beds? So it's just a matter of like gap size and what can like pass through it. Okay. That's gonna lead us, we've talked about the arteries over here. We talked about the capillaries. Last thing to talk about are the veins. Now, if you remember, we said that the arteries got smaller and smaller and smaller as we went towards the capillary bed. It's going to do the opposite. The vessels are going to get bigger and bigger as we move towards the heart. Okay? Because they're tiny here, they're going to get bigger. So the companion vessel, or like the opposite vessel to an arteriole, is called a venule. This is the smallest type of vein that's going to take blood from the capillary bed. The venules are going to merge and fuse together to make larger and larger and larger vessels that eventually become veins. Okay? Now, one thing about veins is that look at how far it is in terms of circulation. Look how far it is from the heart right here, which is the pump, right? So if this is the pump that's forcing the blood out, the blood is losing its pressure. So by the time it gets to the veins right here, we're under a very, very low pressure system. Okay? So since this is a low pressure system, the blood is going to start to pool in these areas. So the veins are known as what we call our blood reservoir, or it's like where we hold our blood supply. So if we look right here, this is a graph from a different textbook, but I like it because it just shows, it represents where all the blood is located in our body. We took a poll and counted where is all of our blood. You can see that 10% would be in our lungs, about another 10% is in our heart, another 10% in our systemic arteries, and about 5% in capillaries. 65% of our blood, so more than half of our blood, is located within our veins. Okay? So this is where like most of our blood is basically stored in our body. Now, again, because this is a low pressure system and we're far from the heart, think about like the veins in our legs, for example, right? If we're trying to get blood from our toes and our legs and our feet back to our heart, what force is it fighting against? Gravity, right? So gravity is already wanting to push it down, and at the same time, it's a low pressure system. That blood is not wanting to go back to the heart. It's wanting to pool down there. So what structure did we have in our heart that prevented backflow? Valves. We're going to have the exact same structure in our veins, like periodically spaced throughout the vessel, to also prevent that backflow. Okay, so I want to point out that only veins will have that valve, because arteries are a high pressure system. They're very close to the heart. They're not going to need those valves. Okay, so if you see a valve within a vessel, it automatically makes it a vein. Okay. Now, another difference between the arteries and veins is the lumen. Okay, so this was written in the previous slide somewhere, but whenever you hear the word, excuse me, whenever you hear the word lumen, it means like hollow center of whatever organ, right? So hollow center of your stomach, or hollow center of your blood vessel, or hollow center of your heart, whatever. Like the middle, like inside of that organ is the lumen. The lumen of a vein, because it is holding so much blood, is going to be much, much larger than the lumen of an artery. Okay? So I think I actually have a picture. Yeah, right here. Can you guys see how the middle, like the white part, and those are blood cells in there, the center of this vessel is much smaller than the center of this vessel, right? So this right here, just based on size, is your vein. Now, if you're like, okay, well, I can't tell if one lumen is bigger than the other, another thing to look at is the tunica media, okay? The tunica media of an artery, because it is in a high pressure system, it has to withstand forces, right? So the tunica media in an artery is going to be much thicker than the tunica media of your vein. So this is the arteries are gonna have smaller lumens, but they're gonna have thicker walls, okay? And you can see that right here. The tunica media is like this highlighted blue part right here, and the tunica media on the vein is the highlighted blue part over here, okay? Now, third thing that's different is that an artery will usually maintain its like round structure. So you can see right here that is relatively circular. Because the veins are in a low pressure system, they're gonna kind of collapse, right? So you can see right here that it's lost that round structure and it looks almost like flattened right here, okay? So it could be in any shape, like if this was your vessel right here, then whatever like is happening to the vessel, it could be like flattened like a pancake, it could be folded, it could be twisted. So the shape of the vein changes depending on like the surrounding structures and like the situation at the time. Does that make sense? Any questions on the differences between arteries and veins, or vein structure? So let's talk about some homeostatic imbalances. There's a condition called atherosclerosis where your vessels become what's it called? Rigid, okay? And the reason is because the tunica media basically starts to allow fats and plaques to build up in the vessels. So think about your elastic in your muscular arteries. They're supposed to stretch and recoil, vasoconstrict and vasodilate. They're supposed to be able to like change their diameter, right? If you fill this up with fat instead, fat is like a hardened structure. It's going to lose its elasticity, okay? And at the same time, if you have enough fat, if you look right here, it builds up and builds up. You can see that it's going to narrow the lumen of your vessel, and now your blood cells are going to have a hard time coming through here, and you're going to have decreased oxygen delivery to like the different parts of your body. Okay, so this is specifically like atherosclerosis. The word like sclerosis means like hardening of the vessel. It's usually due to fats. It could be due to other things as well. But you can see right here that this says atheroma. This is that plaque buildup. Okay? So the way that they usually treat this is obviously first they're going to recommend diet and exercise just like everything else, right? But if it gets to be a condition where diet and exercise is not going to fix it, you have to fix it surgically. What they do is they put in what's called a stent, okay? So if you look right here, the first thing that they do is they stick a balloon into the vessel, which kind of widens it up. So up here you can see that the lumen is very small. It's basically putting in a pump and forcing that vessel to widen, okay? Once that vessel widens, they open up and stretch out. It's like, think about like those like collapsible balls, right? The wire collapsible balls. When you widen it up, that's what this is doing. You're basically taking that wire mesh and holding that vessel open. Then they can deflate the balloon, pull out the wire, and what's left is that cage. And so it's just artificially like holding up the vessel walls because your walls are too weak to do it themselves. Okay? Any questions on atherosclerosis or how to treat it? Yes.
Is that like a temporary fix or does it
Permanent.
The problem?
It's permanent.
Yeah.
It depends on like, they usually do this in your coronary arteries, especially like if you have like plaque, like you're in danger of having like heart attacks. So this is not something that they're just gonna randomly do in any vessel anywhere. It has to be like basically severe enough, or it has to like occlude a vessel like enough that like the blood can't get through. But yeah, this is definitely permanent. Yes.
What if more plaque builds up around the wire? Like, do they just remove it or do they shape it? What do they do?
They could probably go back in there endoscopically and scrape it, but they usually like have chemicals also within this wire that like try to help prevent plaque buildup. It's kind of like your vessels like automatically secrete a chemical to prevent platelets from attaching to the walls. They secrete like prostacyclin and all these other things. So they secrete something similar to prevent plaque buildup here. Other question? Okay, so atherosclerosis is a buildup of that vessel wall and hardening of that vessel wall. An aneurysm is when the pressure in your vessel wall gets so high that it starts to tear that inner, like that tunica intima, okay? If we tear the tunica intima, which is the innermost layer, blood is then gonna start pooling in the tunica media layer. Does that make sense? Okay, so basically we're saying that if we go back to this right here, if we tear this inner layer, then now we don't have that protective barrier. Blood is gonna start to pool in this middle layer right here. If blood is pooling in this middle layer, that's gonna like, it's like taking a balloon and like blowing it up, right? It's gonna stretch it and stretch it and stretch it. If it stretches, the aneurysm, I wanna point out, the aneurysm itself is not what's dangerous. What's dangerous here is the threat that you can pop this aneurysm. And if this ruptures, then now you're gonna be bleeding internally, okay? So, whenever like a doctor does a scan and they're like, oh, you have an aneurysm, it's not like dangerous itself to have an aneurysm, it's the danger, the fact that it can rupture, okay, especially in high pressure situations. So the most common places that you get an aneurysm, this right here is showing you your abdominal aorta right here. So you have abdominal aneurysms, and you also have them in what's called the circle of Willis in the brain. So remember how we said that like if you occlude the coronary artery, it's a heart attack, right? If you occlude an artery in your brain, it's called a stroke, okay? And an aneurysm in the stroke, or an aneurysm in the blood in the brain, is also a stroke, which can lead to a hemorrhagic stroke where it ruptures, okay? So the brain and your abdomen are the two most common places that this happens. Any questions on this? The venous homeostatic imbalance is varicose veins. This is usually a disruption to the valves in your veins. So we said that like if you have a vessel right here and your heart's up here and your blood is going against gravity, we have these valves right here that act as checkpoints to prevent the blood from going back in the opposite direction. If this valve, let's say that this valve right here breaks, it's broken now, then now the blood here is gonna pool and it's gonna come back. What's gonna happen is this vein right here is gonna start to dilate and dilate and dilate more and more because more blood is pooling in that area, right? So if this blood vessel is dilating, it's going to, especially at the surface, become visible because it's going to start to fold or pinch on itself because it's so dilated. This is usually happens in cases of prolonged standing. Okay, so if you are standing for a really long time, then obviously gravity is trying to push the valves down. They can only work for so long until they start to break, right? It also happens in obese people because if you're obese, all of the fat in this area is basically compressing on these veins and it's adding extra weight, so the blood is not wanting to go back up. It's gonna start to pull back down in the lower leg area as well, okay? So, and it also happens in a lot of times when you are standing still, not standing, but sitting for long periods of time. So think about long plane flights, right?